morning, students. Today we are going to start a very interesting and first chapter of plant physiology, that is photosynthesis in higher plants. Photosynthesis is an enzyme regulated anabolic process which is responsible for manufacture of organic compound with the help of carbon dioxide, water and sunlight. It is a physico-chemical process by which green plants and certain other organisms use light energy to synthesize the organic compound. Photosynthesis is important for two reasons. It is the primary source of food for all the living organisms, as well as it is the only process responsible for release of oxygen into the atmosphere by green plants. So here are certain early experiments related to the existence of the process of photosynthesis. Joseph Priestley in 1770 performed a series of experiments with bell jar, candle and mouse. He observed that a candle burning in a closed bell jar soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damage the air. Here, damage the air means they are deteriorating the quality of air by adding the carbon dioxide in high concentration into the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. By this experiment, Priestley hypothesized that plant restore to the air whatever the breathing animal removed by the process of respiration and burning candle by the process of combustion. So here is the diagrammatic representation of Joseph Priestley experiment. It is clearly visible that in first bell jar, in bell jar A, mouse is also alive and the candle is also burning. But in bell jar B, mouses die and the candle also extinguished. Similarly, when he kept the mint plant in the bell jar C, both of them are alive. That means the mouse is alive and the candle remains burning. So he concluded that whatever the air or the carbon dioxide exhaled by mouse or the burning of the candle is taken up by the plant by some process. In this series of experiments, Jan Ingenhousz in 1730 showed that sunlight is essential to the process that somehow purifies the air fouled by burning candles or breathing animal. He performed his experiment with an aquatic plant and showed that in bright sunlight, small bubbles were formed around the green part of the plant, while in the dark they did not appear. Later he identified these bubbles to be of oxygen. Hence, he showed that it is only the green part of the plant that could release oxygen. Julius von Zach showed that the green substance chlorophyll is located in spatial body known as chloroplast. He provided evidence in 1854 for the production of glucose in the green part of the plants and stored in the form of starch. Another experiment is done by Engelmann in 1843. He split the light using a prism into its component parts and eliminated a green algae, cladophora, placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of oxygen evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the spectrum. He first described the action spectrum of photosynthesis. The action spectrum resembles roughly the absorption spectrum of chlorophyll A and B. The last experiment in this series is done by Cornelius van Niel in 1897. He conducted his experiment with purple and green sulfur bacteria and demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a hydrogen donor reduces carbon dioxide to carbohydrates. He gave the present-day equation of photosynthesis. In green plants, water is the hydrogen donor and it is oxidized to oxygen. Purple and green sulfur bacteria use H2S and the hydrogen donor, and so the oxidation product is sulfur and no oxygen is produced. Thus he inferred that the oxygen evolved by green plants during photosynthesis comes from water and not from the carbon dioxide. This was later proved by using radioactive isotope of oxygen. The overall correct equation for photosynthesis is as follows: 6CO2 plus 12H2O in the presence of light, C6H12O6 plus 6H2O plus 6O2.